pro-organic agriculture, and pretty much again plant biotech. But over the years, I've come to realize that it's a bit more like plant biotech and organic agriculture can actually evolve together instead of against. And now I'm trying to encourage this mentality and this way of thinking that we should be talking and communicating between our two communities instead of trying to just be against each other. So, like Louis said, I'm a prof at McGill University, but I also graduated at McGill. So I did my master and PhD in plant science at McGill University and also at Serum, which is a research center dedicated to field crops that is located at Saint-Mathieu-de-Belleuil, a bit outside of Montreal, the island. I'm a course lecturer, so I've taught 2 courses at McGill, so experiments in biotech and plant physiology. And I'm a recent postdoc. I'll be starting October 1st at CNRS, and I'll be talking a bit about that. So during my master degree, I studied the circadian clock in rice. So we have a circadian clock. Plants have a circadian clock too, and this is what I studied. I've studied the molecular mechanism that are guiding different responses in rice. During my doctoral degree, my objective was to find new genes that are associated with early maturity in soybean, which is related to their circadian clock. And I was using different technologies such as CRISPR-Cas9 to edit the soybean genome to validate the function of those genes. Now for the CNRS in Montpellier, as you may know, Montpellier it's not Montpellier in Vermont, it's Montpellier in France. So I'll be starting there in October. CNRS is one of the biggest research center in the world. It's actually one of the best. It's for plant science and agriculture is ranked at 4th. And Montpellier is actually the biggest and largest hub for plant science, hosting more than 1,600 researchers that are working in the field of genetics, molecular mechanisms. And it's ranking pretty high in all different ranking system, including the CIMAGO ranking system for societal innovation, research, and just overall in general. So CNRS, it's a big deal. I'm quite proud to be to have been accepted to work there, and I'll be more than happy to try to bring some of CNRS inputs to Quebec and McGill and everyone here, just because I think it's an exciting opportunity. So at CNRS, I'll be working on white lupin. White lupin is an interesting legume, and I'll be using white lupin in my research at CNRS as a template to understand plant biotech in general. I'll do so because this is what I know. And I think it's always better to talk about something you know than something you don't know. So I'll use that as a template to explain to you and guide you in the world of plant biotechs. So at CNRS, I'll be working on white lupin. White lupin is a really interesting plant because it forms something that we call cluster roots that uptake phosphorus. It's one of the only cultivated plants that do that, and it's really interesting. And our role and our objective is to map the genes that are involved in this cluster roots development. And to do so, we're going to use different types of biotech, and later on what we want is to transfer those genes to soybean, like a commercial field cultivated plant, and use genetic engineering to do so. So this is our overall objective, and we have a big pipeline to achieve that goal. And yeah, that's pretty much it. So yeah, like I said, to make this presentation a bit more sound, I'm just going to use example that are relevant to my future postdoc project. And if you have specific question about that, I'll be more than happy to answer them. And yeah, that's it. So this is the overview of my project in France. It's pretty big. It's pretty overwhelming. So I won't go over each and every detail, just to tell you that a postdoc project is a big endeavor, but I'll use that as a template to explain basically what are biotechs. So during my during my postdoc, what I'm going to be doing 5 types of biotech. It's not like they're not strictly pure biotech. Sometimes they're kind of biotech adjacent, but people in biotech need these skills or type of experiment to be able to achieve their goals. So the first one is high throughput phenotyping. I'll explain that a bit later on. We're going to be using bioinformatics and in silico predictions using different types of sequencing. We're going to be doing some synthetic biology, so cloning, generating new cloning vectors. We're going to be doing some genetic engineering using CRISPR-Cas9 and other form of genetic engineering. And we're also going to be doing a bit of conventional breeding, which are not biotech, but most people in plant biotechs are using some form of conventional breeding to be able to achieve specific objectives. So 5 different techniques, and I'll go over each of them pretty quickly with some examples. So high throughput phenotyping, really interesting field of study. It's evolving extremely rapidly, especially with AI. So basically, it's a method used in plant and animal science to measure a large number of physical, biochemical, and physiological traits, what we call phenotypes, using basically automated technologies. So sensor, drones, robotics, and like I said, it's a rapidly evolving field. People use HTP, so high throughput phenotyping, at different scales. So you can use it at the cell scale. So for example, if you're modifying cells or engineering cells with some types of fluorescent protein, and you want to phenotype those cells in an automated fashion, automated manner, you can also phenotype or high throughput phenotype different plant or organs. So leaf, seeds, roots. In my case, I'll be phenotyping roots for my project. So we're right now developing a high throughput phenotyping pipeline to be able to take a lot of measurements pretty quickly. And you can also high throughput phenotypes plants at a plot or field level if you have the technologies. So at the cell scale, I'm not doing this, but a lot of people are implementing ways to phenotype at the cell level using some form of robotic devices. So you'll be using fluorescent protein, you'll be modifying cells in different fashions, and you're going to be phenotyping the response of the cell using different mechanisms or different devices. In my case, for my project, I'll be using a CT scanner to scan the roots of my white lupin plants. So what we're going to be doing is we have a population of white lupin. It has more than 150 accessions, so 150 lines, and we're going to be using a CT scanner to measure the properties of the roots and how the roots develop. So we just scan the roots exactly like you would scan like a human brain for tumor, for example. And using this, later on, we're going to be using some form of software to analyze the root development and the root properties. So we're going to be analyzing different traits such as root length, surface area, like width, depth, and number of roots. And using this, we're going to be able to infer different type of information that I'll be explaining a bit later on. As you can see here, with CT scanning, you can also generate 3D images, which is kind of interesting because now you can see how the plants evolve in a 3D fashion, which was not possible before. And as you might guess, phenotyping a root system that is like evolving in a natural medium, so soil, something like that, is a really hard task to do. And CT scanning provides us a means to do so quite easily compared to what was done before. So before, people were basically washing roots and trying to not break the root particles or the rootlets, and it was kind of cumbersome and not really precise. Now with CT scanning, we're able to do this kind of work more rapidly, more efficiently, and more accurately. You might be interested in this. I'm not using this technology, but a lot of people are implementing drone-based measure and high throughput phenotyping. And you can use drone to measure a lot of traits and with a lot of types of images, and yeah, basically a lot of type of images like multispectral, colored images, thermal images. And you using this, you can basically measure at a field level or a plot level how a crop is behaving or how a specific line is behaving. I'm not doing this in my project, but I'm one of my friends that did his PhD at UQAM, so l'Université du Québec à Montréal, has started a company based on his PhD project. So he was working with drone, taking a lot of measurements, and now what he's doing is basically helping farmers to locate specific problems in their fields. So they're scanning or they're sending out the drones to see if there's insect infestations, or then sending out the drones to see if the land is well drained, if there's some problem with the land, if there's pathogens problem that are located specifically in some pockets in the field. So I think it's interesting, and it's a kind of a clear example of what you can do after a PhD. You know, during a PhD, you learn specific skills that you can apply later on, either in public institution, private institution, or public-private institution. In his case, he started his own company. It's working pretty well, and it's getting traction in the farmers community, because a lot of farmers, what they have is they have big tractors and everything, but something they lack is time. And with drones, they're able to save a lot of time, because you can fly over drones over their fields, and they will not have to scout all of their fields uh, by foot, basically, by walking there and trying to locate the specific problems. So this is a kind of clear example of what you can do, and I think it's pretty cool. And since it's my friend, I wanted to present that. So in silico and bioinformatics, you might have heard about this in the media. A lot of genomes getting sequenced nowadays. The human genome has been sequenced decades ago, and this is getting a lot of traction because now sequencing is affordable. It's more than affordable. People sequence the DNA of their dog, their own DNA, and they are sending this to to companies that are just processing their own genome or the genome of their dog. I'm not going to discuss about the details of this and everything, but it's now pretty affordable. And as scientists, we're using a lot of these data sets, sequencing data set, to understand how plants are working and what are, I would say, the genetic differences that are making up these different traits. So our goal with sequencing is to find new variations that are coding for specific traits. In my case, it is for cluster roots development. So what we want to find and identify is new genes that are coding for this. Is this like a basic question of ours is this gene coding for cluster root development? And would it be possible to transfer it to soybean? And sequencing the genome of white lupin and soybean is a way to pinpoint and identify some of these candidate genes that could be implicated or involved in the regulation of this trait. And using
Can everyone hear?
No, I think the audio has been like muted in some way. We can't really hear anymore.
Wait,
Oh,
wait.
really?
Oh no, it's
Oh,
back. It's
now
back.
now we now we can hear you. Yeah.
Okay, okay,
Okay.
perfect, perfect. Thank you.
Actually, I think my internet connection is unstable. I'm just going to move to another room. Just give me 15 seconds. Sorry about
Thank
that,
you.
guys.
Thank you.
Did you miss a lot or?
No, just around like brief, like 15 to 20 seconds.
Okay, so I'll just repeat to make sure everyone understands this part. So sequencing is not a very affordable technology. People can sequence their own DNA, the DNA of their dog. I've sequenced the DNA of my dog because we didn't know what breed she is actually. And it's pretty affordable. And as plant scientists, we're trying to harness this affordability to answer our question. In my case, we're trying to identify the genes that are involved in cluster root developments and white lupins to transfer this trait to soybean. And to do so, what we're doing basically is sequence a lot of white lupin accession, so a lot of white lupin lines, and compare it to the soybean genome to try to locate what we call candidates or candidate genes that could be involved in the regulation of this trait. And now with the power of AI and computer and software, you can predict the effect of some mutation. So you don't even have to run experiments to have a lot of information about specific genes and specific mutation. So I would say nowadays, most scientists use these computer-based methods before entering the lab. It's just kind of a pre-test before doing a lot of costly and expensive experiments. So you use these methods, you're trying to limit or decrease the number of candidate genes by using these methods. You're basically trying to find the 2, 3, or 4 candidates that would be the best for your future experiments that are going to be a bit more hands-on. So it's really practical. It's really like it's computer based, but I would say most scientists use this nowadays because computers are so powerful and it's much uh, much more affordable to have someone play on a computer and get this information through the lab instead of paying them to do things that are a bit random, I would say. So bioinformatics, so with DNA sequencing, there's a lot of different steps. You might be involved in of these steps depending on your lab and depending on the company you're working with. But I would say most people nowadays, what they do is they extract DNA and they're going to be sending the DNA to a DNA processing facility or a sequencing processing company. So us, we work with two different companies. One is Genome Quebec, and the other one is IBIS, located at Université Laval, because those guys have developed specific protocols for specific application. So we extract the DNA, we send the DNA to them, and they're going to be fragmenting the DNA using different types of enzyme. They're going to prepare the library. They're going to be doing the sequencing, and they're going to send back to you the results of the sequencing. From there, it's only the first part has been done. Now what you need to do is process these data sets to have real information that you can use. And this is what we call bioinformatics because you're going to use the files. So what I mean by the files is actually this, and you're going to be processing all of those data sets to be able to get actual sequences that you can use to understand your question or find answer to your main question. So when you're doing bioinformatics, you're going to be dealing with a lot of files that look like this. I'll be honest, it's not very exciting when you look at this way. It's like working in lab looks much more exciting, but it makes your work in the lab so much more efficient that almost everybody develops some kind of bioinformatics skills nowadays. So if you want to do plant biotech, you're going to be dealing somewhat with this. So, like I said, what we want to do is find the letter. So what I mean by the letter is the nucleotide or what we call the base. So in the genome, you have 4 different bases, A, C, T, G, and you want to find one or multiple letters that change the phenotype. In our case, this letter is going to be involved in cluster root development. So if you look at this, it's just an example. Here you have the human genome, but it could be this letter that could be involved in the cluster root development if it was for the white lupin genome. So you want to find this letter that changes everything and that, for example, increase the number of root for your white lupin. So like this gene, like you'll have a much better idea that this gene is involved or might be involved in the cluster root development if you see an association between this letter or a statistical association between this letter and your trait. So using bioinformatics and in silico prediction, what you can do is basically you can do some gene mapping. I would say I'm a specialist in gene mapping. I've published many articles about this in soybean. You can also use this technology or sequencing technology to be able to do genomic prediction. So genomic prediction is basically you're able to predict what are going to be the best combination. So in plant biotech and plant breeding, you're basically often crossbreeding 2 parents. So what we call the parent line. So you use mama and papa and like you crossbreed them. And what you want to do is find the best combination to get higher yield, best disease resistance. And using sequencing and bioinformatics, you can infer pretty reliably what are going to be those best combination. So what it makes in the end is that you're going to basically remove all the superfluous or inefficient combination, and you're going to be focusing on the one that are predicted to give the best results. And it's pretty efficient. They're using this in the dairy industry. They're using this in plant breeding for a lot of different plants, including soybean, the plant I was working on during my doctoral degree. And we're using this at Serum. And honestly, it's quite magical. It's working extremely well. And a lot of those companies that you see, those big companies, but also smaller companies, they're reorienting their breeding programs toward a genomic prediction approach. And in addition, you can also use marker-assisted selection. So one of the problems with plant breeding is that a lot of the plants that you're going to be breeding or developing have traits, or you would like them to have traits that are not easily phenotypable, if I can say. So, for example, one of them is resistance to soybean cyst nematode. Soybean cyst nematode is a kind of extremely small worm, almost invisible to the eye, that are present in fields, and it's extremely difficult to find plants that are resistant to this very efficiently. But what we found, because we've used gene mapping, is that you can use the information from the gene mapping to find genes that actually code for the resistance to this pest. So what we do is we find the genes that are involved in this, and we only use markers to find the plants that carry on the genes that provide resistance to soybean cyst nematode resistance. You also have this for different diseases, and it's kind of a affordable, easy way to circumvent phenotyping plant for different like very challenging phenotypes like disease resistant and pest resistance. So people now also are kind of reorienting their all breeding program to include marker-assisted selection. So with sequencing technology, you can do all of this stuff and so much more. And what you can do is that you can integrate high-throughput phenotyping data to sequencing data to actually find genes that code for specific traits. And using this, you're going to be using different types of bioinformatic method like genome-wide association studies or QTL mapping to find the specific genes that code for your trait. So after you find an interesting candidate gene using high-throughput phenotyping and sequencing technologies, you need to validate the function of this gene, because at this point, what you have is only predictions. You have predicted that this gene might be involved in the regulation of this specific trait, but what you want is a final validation, and with a final validation, you can now use the genes for different things. So to achieve that, people now use synthetic biology. You might have heard about this at your high school or CJEP or university if you're a university student. It's a rapidly evolving field also. So it's a rapidly evolving field that use engineering principles to create new biological components. Most people refer to synthetic biology as cloning. It's a bit larger than cloning, but I would say overall, like cloning, creating new vectors, creating new plasmid is all part of synthetic biology. So like I said, synthetic biology makes an extensive use of gene cloning. So gene cloning is the process of making identical copies of a specific gene or DNA segment, and you use this to generate a vector. And using your vector, you'll be able to do a lot of different things, like creating GMOs or producing proteins. So synthetic biology, I'll give you just a few examples of what you can do using synthetic biology. So here we see different plants, probably tobacco, that have been transformed with a marker or a gene that is called ruby. Ruby comes from the beetroot, and it gives this red color to plants. And what scientists have been able to do is that they've cloned the ruby gene. In this case, it's 3 different genes from beetroot, and they're now able to integrate these genes in other plants like soybean, tobacco, and now these plants are producing this compound called beta-laine, which is good for your own health conditions. You have different marker like the BX marker that comes from the yellow beetroot, and instead of providing a red color to plants, it provides a yellow color. And finally, you have other markers like chromopy that gives pink hue to different plants. So this is something that can be done using synthetic biology. What you can also do with synthetic biology is insert a fluorescent protein like a green fluorescent protein or an M-cherry. So if you look here, you see places that have been genetically modified to code for a fluorescent protein. And these leaf tissue are now fluorescent using an M-cherry marker. So people use this to track how genes behave in a plant, or like this is one of the applications, but you can use this to really track, okay, where is this gene located? What does it do? Does it migrate? When is it expressed? And it's really interesting. It's a kind of convenient way to see and understand how gene behaves. You can also use M-cherry to understand where you've introduced other genes. So in this case, they use the M-cherry that you see here to understand that they've introduced another genes along with the M-cherry, and that it's located now in the leaf and the shoot of this plant. And in the end, when the plant is going to flower and generate seed, you see that the seeds also carry the M-cherry gene. So it's really interesting. Now the plant has integrated fully the M-cherry marker. So it's quite interesting. And you can also use synthetic biology to generate herbicide-resistant plants. So what you can see here is plants that are herbicide-resistant because they have been transformed, or they have been, transformed, this is the right word, with an herbicide-resistant gene. So when you apply a specific herbicide like glyphosate or glufosinate, or imazapyr, or all of these herbicides, now your plant is going to survive. So it's quite interesting. I'm not especially pro herbicide, but it's kind of a convenient way to study specific genes. So synthetic biology, you can use synthetic biology to produce proteins of interest or different enzymes. You can create GMOs like the one you see commercially, but also GMOs for fundamental studies. You can use synthetic biology to modify, insert, or delete genes of interest. And you can use synthetic biology to study the function of specific gene. And in my case, I use synthetic biology to study specific gene function to solve fundamental question about cluster root development. So the ideal method of confirming a gene, I would say the staple nowadays is to edit the gene or do some genome edition with a technology called CRISPR-Cas9. So if you've been watching the media or you're basically going to school or CEGEP, you might have heard about CRISPR-Cas9, especially if you're at this webinar, because I would say nowadays everyone has heard about CRISPR-Cas9. It's now kind of a staple in the industry and like biological studies. But I'll just go over what it is because it's really interesting. CRISPR-Cas9 in plant biotech can be used to do so many different things. As you can see here, you have just a wild type tomato. So this is the tomato you will find at the store. And scientists have used CRISPR-Cas9 to edit the genome. So what they've did is they've modified 3 different genes that are involved in the synthesis of different pigments. And by doing so, they've been able to first confirm the function of these genes in the pigment biosynthesis, but also generate a palette of color for tomatoes. So now you have orange tomato, yellow tomatoes, green tomatoes, and all of these tomatoes are ripe. They're just as ripe as the red one. And commercially, this is appealing because now you can use a pretty cool tomato, and instead of crossbreeding it with another variety that might already be yellow, you just have to edit the genome, and in 1 year, you generate this palette of color. And for example, if you want to sell this red tomato but want to create a really looking good product, now you can generate this palette of color, farm this, put this in a box, and people will be more than happy to see kind of this palette of color. So this is just a cool application of CRISPR-Cas9. Also, scientists in the US have been able to prove using CRISPR-Cas9 that different genes are involved in yield, like getting an increase of yield. So what they did is they managed to modify 3 different genes that are called FFT, SSP, and SPSP. So they've modified these genes to generate higher yield for tomato. And this is quite impressive and interesting. And this is going to be my last example about tomato. Some teams in the US, they've been able to modify only 1 gene and increase the tomato size drastically. So they've been able to confirm and validate the function of this gene CLV3, and they've been able to link and associate CLV3 to fruit size. And what you can see here, this is a wild type, so the natural tomato that you would find in the wild. And by modifying CLV3, they've been able to recreate your typical beefsteak tomato that you buy at the store. So this is a really powerful technology and extremely interesting for plant scientists because first, it's precise, you save time, and you don't have to go over all of this crossbreeding work that takes years to accomplish. And to do CRISPR, like to do some genetic engineering, you basically have 2 different steps. You have the synbio, so synthetic biology, and you have the genetic engineering steps. And you cannot do you have to do both at the same time, you cannot do only one. So if you do the synthetic biology, you'll only have vectors without results for your plant, and you cannot do genetic engineering if you're not doing synthetic biology. So to do so, you have to go through a step that is called plant transformation. Most people that do plant transformation use one technique, which is called transformation with Agrobacterium tumefaciens, but you have so many different techniques. And Agrobacterium tumefaciens, I would say this is kind of the staple in the industry. It is a bacteria that can be used to deliver desired gene into your plant genome. And this is a widely used technique to create GMOs. And the bacteria naturally has the ability to transfer gene to plants. And what scientists have been able to do is that they harness this feature to be able to transfer their genes, so their genes of interest to plant, not the genes that the bacteria want to pass, but their genes. It's a very clever way to transfer something very specific. And how you basically do this, you wound the plants using different methods. Here they prick the tip of the plant using a needle, and you inoculate this injured organ with your bacteria. And the bacteria respond to, so is it okay? It's okay, perfect. Okay. And the bacteria respond to plant wounding. It's attracted by plant injuries because what they want to do is invade the plant and pass on those genes. This is like their natural mechanism of survival. And by doing so, you transfer a protein that is called Cas9. And Cas9 is kind of the second part of CRISPR. CRISPR is the method or the mechanism. It's called cluster regularly interspaced short palindromic repeats. We'll say CRISPR, much shorter, much more, much easier to say. And it's part of a natural mechanism that allow bacteria and protect themselves from viruses. And what scientists have done is they've used this mechanism and harness the interesting part to perform genetic engineering. So they've used this feature from the Cas9 protein, so this immune system feature, they've modified it, and they're now use it to do some genetic engineering with plants to inactivate or activate or do a lot of different things with specific genes. And now the inactivated genes can be studied. And this is what I'm doing in my research. So there's three main steps to genome editing. So you're going to create a piece of RNA. So you're probably all going to high school, at least CEGEP or university, you know what RNA is. And this piece of RNA is going to match the target DNA sequence. So here you have your gRNA and it matches the target DNA sequence. This RNA is going to guide the Cas9 protein. So here you have Cas9. And what Cas9 is going to do, it's going to cleave the DNA right at the spot where you want it to cleave. And by doing so, it's going to introduce errors in the DNA sequence. And what you want is errors to be introduced. By introducing errors, you're going to really modify completely the DNA structure, and you're going to be able to understand what this gene is actually doing. So as you can see here, and it's just an example of how you can use it in research. This is soybean. Soybean generate naturally nodules, these small balls. And here you have just a wild type, you're controlled, a non-modified plant. And by modifying different genes, you can increase the production of nodules. And nodules are interesting because they fix nitrogen. Nitrogen is a plant nutrient and it's quite expensive, but soybean has a natural ability to fix the nitrogen that is located in the atmosphere. And now you can generate more nodules that can fix more nitrogen. So you're going to save more fertilizers by knocking some genes, by inactivating some specific gene. So a very clever way to save on fertilizer cost. So like I said, CRISPR-Cas9, the goal is to introduce specific mutations. So the red lines you see here, those are those specific mutations that have been introduced using this protein. Pretty cool. And after this, you're going to be using sequencing. So the in silico and bioinformatic part that I was talking to you about. So you're going to be using that to identify the mutation that you've introduced with the Cas9 tool. So I said a lot of words, like I'm pretty chatty, as you can see, but I'm just going to do a recap about genome editing. So you can use it to improve crop faster. Just to give you an example, when we generate a new variety at Serum, it takes about 10 to 15 years, but with Cas9 and genome editing, it takes less than 2 years to get a final variety. So it's much faster and it's much more accurate. Because in the end, you're going to be generating a few plants, while conventional breeding, you're going to be generating hundreds, if not thousands of plants that you're going to need to screen in fields. It's very complicated and lengthy. With genome editing, you can introduce specific disease resistance to increase crop yield. You can use it to make GMO with different, like increased nutritional values, and you can do so more quickly and accurately. You can increase crop tolerance to environment, and you can use it to study plant genes, just like what I'm doing. Actually, genome editing is much broad, like its scope is much broader. I'm just giving you a few examples because we don't have 3 hours to talk about all of the different possibilities that like are made accessible by genome editing, but you understand the principle. I wouldn't say it's a magical tool, but it's quite novel and mind-boggling in comparison to what we were doing before. So conventional breeding, this is not a biotech, and I've been talking about conventional breeding during this talk. It's not a biotech, but most people still do it. Why? Because it's kind of reliable. These technologies, although Cas9 has been around in plants since 2013, they're still fairly new. It's not all plants that you can use Cas9 efficiently and very easily, and it's still fairly new. Conventional breeding, it's kind of a staple, and it also integrates the information from the 4 other techniques to generate generate new cultivars. So most breeding facilities and most labs still use some part of conventional breeding in their pipeline. This is just how it is. So it's not a biotech, but you'll encounter conventional breeding if you're interested in plant biotech. And like I said, conventional breeding, nowadays, it has evolved. It's still fairly slow, but it incorporates information from different fields. So in conventional breeding, you're going to be using genome prediction and marker-assisted selection, so bioinformatics. You're going to be doing some statistical analysis, doing high-throughput phenotyping to locate the best performing plants and the plants that have specific features that you want to integrate in your new crop. You're going to generate new vectors to use it with different technologies, so something like CRISPR-Cas9. And you're not only going to generate one specific plant with CRISPR-Cas9, often you're going to be combining different plants that have different traits generated with CRISPR-Cas9 using conventional breeding. So what I'm just saying is a lot of people dismiss conventional breeding because it's kind of old-fashioned, but actually people still use it, and it's still part of the pipeline, the biotech pipeline, although it's not a biotech per se. So this is just what I wanted to highlight. And here you can see some of our plots, some of Serum plots that are cultivated right now in New Zealand. So it's quite magical. What you see in red are our plots, and we grow these different cultivars in different environments to see how they're going to respond. Because when we're developing a cultivar, we're sending this everywhere to see if it's responding well, because we're going to be like what we want to do is commercialize this globally if possible. So right now they're doing pretty well, and it's quite interesting to see that something that has been bred in Quebec can do well in New Zealand. So in conclusion, like I said, I'm pretty chatty. I hope you enjoyed the presentation so far, but I'm just going to try to conclude. And after, I hope you have some questions for me, because if you don't have questions, I'm going to be asking you questions. And yeah, this is just how it is. So the title of this presentation was Plant Science and Biotech, Solving Recurring Problems with New Approaches. And I just want to conclude with the fact that novel biotechnologies are new avenues for crop improvement, and that they open up new area for research. Imagination and innovation are the work horses of this exciting field of study. And the things that our plant biotechnologists do, like regularly or daily, depending on what are like their job titles. So they're doing a lot of experiments. They're going to go to their lab, of course, but also in fields, and going to be doing a lot of computer work. It's a multidisciplinary field of study that not only include your team and many members in your team, but also different teams from elsewhere in the world. Like us, like in France, we're dealing with, like, we're located in Montpellier, but we're actually working with people from Denmark, people from France, people from United Kingdom, and soon probably people from Quebec. So pretty cool. You're going to be attending different symposiums and conferences, and you're also going to be presenting at these symposiums and conferences. You're going to be writing articles. You're going to be reading a lot, doing some literature reviews to stay in touch with new developments. You're going to be networking, and I highly emphasize networking because it's kind of this is kind of important and often overlooked. And you're going to be traveling a lot to meet new people, learn new technologies. You're going to be supervising people. So even if you're a technician, you might be supervising undergrads or students that are doing internship during the summer. And if you're a researcher, you might be supervising a whole team, but the chances are that you're going to be supervising someone at some point, and you're going to be doing maybe some grant application to get funding for your research. If you're not doing grant application, you're going to be working with a selling team that is maybe selling a product. But what I want to say is that you're going to try to find a way to find some funding or money to be able to do your research. Maybe you're not going to do it directly, but in some way you're kind of be doing it. Us at Serum, we do a catalog of cultivars where, like, we need to meet officials that are involved in certifications and providing certificates that our crops, like our new cultivars, are much better than our old one and stuff like that. So I would say it's paperwork, all in all, grant application, but also other things. The qualities for plant biotechnologies, sorry for the S, there's a mistake here, small typo. So first thing is perseverance. Like, you're not going to become the best scientist in the world in a day or a month. This is a really expanding field. There's a lot of different plants, a lot of different organisms, and the thing you need first is perseverance. A lot of smart, very, very smart people have abandoned pretty quickly because they were not perseverant enough. You're going to be failing some experiments at some point, probably during months, maybe weeks, or even years. You're going to, like, you need to be able to do multidisciplinary work with a lot of people. So you need to be flexible. You need to be able to do computer work. You need to be able to go to the field. You need to be able to work in the lab. Maybe you'll only work in the lab and not do the other things, but you must be able to understand what the other ones are doing. You need to be innovative. You need to have bold ideas. People that are not innovative and that don't have bold ideas, they just do not survive in the field. That's just how it is. But this is the same thing for everything nowadays. You need to be innovative. Like our world is expanding rapidly, everything's changing rapidly. People that have the best ideas and that are innovative, they're going to be able to not only survive, but make a good career. You need to have a collaborative state of mind. Like collaboration is key to be able to do all of this stuff because honestly, you cannot learn everything and do everything perfectly. So you need to be able to collaborate with your own team members and members from other teams. You need to be competitive against yourself and not others. This is not easy to do. You still need to be competitive with others, but the main thing is be competitive about yourself. How can I improve myself? How can I write a better article than the previous one? You need to be opportunistic. Surviving in science is all about opportunities, and when you see one, you need to grasp it. You do not wait. You take it. You take your chance, and this opportunity might not come back again. You need to be open to new concepts, technologies, but also way of working. And you need to be hardworking, but also with a touch of fun. So I listen to death metal. I go to death metal show. I stage dive. I listen to gangster rap. I'm trying to be a fun dude, but I'm also able to work hard when it's time to do so. Final conclusion. This is a continuously improving field that need young, innovative mind to find solutions to recurring but also new challenges. And are you one of them? I hope so because I love plant science and I want to recruit everyone that I can. And yeah, thank you very much. And I'm open to question if you have some. Thanks, guys.
Thank you for your presentation. Actually, I do have a question about the CT scanning that kind of caught my eye. I was wondering for CT scanning or the second technology, it was the drone base. I know the main purpose is to kind of like observe the plant from far away, kind of to see, to visualize. But do you think by any chance like that could also work after you like gene edit the crop from far
Yeah, yeah,
away?
yeah. Actually, it depends on the trait and what you're studying, basically.
Mhm.
So in our case, we're going to be using CT scanning at the beginning of the pipeline to try to locate accession, so lines that carry cluster roots and
Mhm.
compare them to lines that do not carry cluster roots. But at the end, we're going to be generating lines that overexpress cluster root genes. So they mass produce cluster roots, and we're
Mhm.
going to be rephenotyping them with our CT scanner.
Mhm.
In the
Mhm.
field, it depends what you're trying to do and depends the gene you're trying to like maybe modify or integrate or delete. So if, for example, I can give you like if you delete a gene that is involved in disease susceptibility, you might be able to phenotype it using a drone-based method, you know?
Mhm.
So you go in the field, you see a crop is slowly dying or it struggles, the color changes, something like that. It becomes a bit more brownish or yellow. You might be able to compare it to a crop that do not change color because it has now some tolerance or resistance to specific diseases. So it really depends on what are your question and what technologies you're using. But what I can say is now technologies have become so efficient and so diverse that I would say it's almost possible to do everything all at once, like depending on what you're trying to do.
Okay, that's good. That's what I thought. I just wanted to verify that. And also within the CRISPR, would you say CRISPR is one of the most used gene editing um technique within the plant science field, or would, for example, single interfering RNA, like gene knocking down be something common? or
Yeah, I won't get into like the GMO debate and like the CRISPR because there's a lot of peculiar details about this, but I would say it's really expanding rapidly. Like all companies now are moving to CRISPR instead of RNA interference and all of these other technologies that were once used, but now kind of like they're not exactly up to the standards of CRISPR. Like CRISPR, like you can edit so many genes at the same time in a precise fashion, specific ways. You can like the difference with CRISPR also is that you can generate non-GMO crop because the Cas9 protein will be removed, so you only keep the edit. So I would say it's kind of evolving rapidly, like the legal field and like the legality of like using CRISPR has been kind of reassured with like the Canadian laws and European laws. So it's legal to use CRISPR for crop editing, but it's kind of evolving rapidly, and we don't really know who's currently using it and to which extent they're using it. So, but I would say yes, like a lot, and I would say in the literature. So when you go to university, you look at like different research station. Nowadays, everyone's using CRISPR. This is just like the staple. So this is kind of it's not a. I hope I answer your question, but sometimes it's a bit hard to provide an answer given the fact that the companies that are using CRISPR, they're not kind of advertising what they're doing. You know what I mean? They're doing a lot of things behind closed doors. So yeah, that's it. But I would like to have some questions from the audience. I don't know, guys, if you have some specific question about like the technologies I'm using or just plant biotech in general, university, are you like high schoolers or do you go to CJEP or do you go to university? Like honestly, don't feel shy. I've been there and I'm pretty cool guy. And if you have some specific question, just feel free.
Hello. I have a question for you.
Sure.
I'm in university. I'm also studying in Mac campus. I'm going to second year right now. My specialization is animal health and disease, so a plant part is a bit new to me, but I do have some questions for you. First is going to be
Sure.
like, how is the plant biology research has been like applied in industries of like or agriculture like in these days? Like how does it like apply to the like to the certain industry or like certain like agricultural like technique that isn't like involved with the research?
Yeah, sure. So I would say it's like it's pretty broad question, but I would say when you look at the like plant research in general, you either have public sector, you have private sector, and you have everything in between. So Serum is kind of in between, but mostly funded by public funds. So I would say there's a big push towards integrating all of these technologies because sadly or not sadly, we live in a competing world, and and governments know that, and they're trying to make sure our industry are up to the standards because everything is evolving so much rapidly. So plant biotechs and plant science is uh, is a bit weird, but all research is weird in the sense that you're kind of competing against the one that you're collaborating with. So I'm competing against people I'm working with in my own lab, and same thing with other labs. So you need to be on point, but you also need to be able to collaborate. And to be on point, you need to have innovative ideas. And I would say like right now, public, private, and private, they're all trying to integrate some form of technologies that I've talked about. So a lot of people are going to integrate, for example, in the private industry, drone-based measurements. This is a really big topic. So because for them, they're really saving a lot of cost. And I would say small breeding companies are trying to implement it, and big breeding companies have been doing it for years. So it's kind of a staple now. For CRISPR, same thing. Small breeding companies are trying to implement the technologies, and big breeding companies have been breeding stuff with CRISPR for years. It's just that they were not releasing it to the market before the legal status of like CRISPR use was kind of like tackled, and you know what I mean? So you still hear me, guys? Yes?
Yes, we can hear you very well. Thank you so much.
Perfect. So, and I would say same thing with public research. If you go to Agriculture Canada or universities, all of these people are trying to implement these technologies. And the main reason for that is if you're really good at doing innovative stuff, you'll get more funding. If you get more funding, you'll get more impact. And this is what scientists want. They want to have impactful science. So, you know what I mean? It always depends where you end up, but I would say this is kind of an objective that all scientists have, is to integrate these technologies, because we don't want to, I wouldn't say we don't want to replicate past mistakes, because they were not mistakes, but we want to improve and do things more efficiently.
Thank you so much. That answered my question. And I have another question for you. I mean, I'm in university and I figured out like by now that what I want to do in my future in my field. But I know that a lot of people in high school and a lot of people in CEGEP a lot of people still unsure about like what they want to do in the future, or what they want to, if the degree they're having is the right choice. So I wanted to ask you, like, what made you like to choose that you want to continue like doing all the way to the PhD in plant science?
And that's a good question. So, like I said, guys, I'm really different. I, I had so many lives. Like when I was young, I was like a militant for different like environmental and social like causes, and like I was really kind of proactive regarding this. After this, I started working as a social worker, working with people with drug addiction. So something really different than science. And you know, when I was social worker, I hated science so much. Like I swear I would never do that of my life. And kind of change it in a way. And you know, later on, I decided to do a technical degree in agriculture, and I really love my degree. But I wanted to move to it like and go to university, just because honestly the salaries were not great, and getting paid like back in the days like $12 an hour or even less was not very appealing to me, especially when you're working like 60 hours a week and you're doing backbreaking work. And honestly, I've found that people in the organic agriculture movement most of them were kind of close-minded regarding technologies and like even using tractors. And I don't know, I wanted to go to university, and when I went to university, I was exposed to so many different things that I didn't know about biotechs, but also different countries and different cultures. You know, like I come from a like the countryside from Quebec, and honestly, I love my place. It's really cool. People are really cool, but it was nice getting exposed to all of this, and I kind of evolved and created my own take on science. Not exactly in organic agriculture, but not also especially in biotechs. I believe in biotechs and I believe in organic agriculture, but I sometimes I find plant biotechs are a bit kind of a bit too technology focused. There are some social problems that needs to be addressed with like social answers, you know, social solutions. And same thing with organic agriculture. Sometimes they should be integrating biotech. So I'm trying to be open and like just help the plant movement, like farmers, plant biotechnologists, students, and get people into plants just because I love plants. I think they're cool. I think like most of our problems could be solved by plants, even like psychological problems and like getting in touch with nature. So it's kind of esoteric in a way, but like this is like I think like plants have so much to offer and just personal like happiness and everything. So I'm kind of like trying to just at least get people to like plant and like if they don't like the biotech like aspect of it, it's okay. But I think plants are pretty cool. So I'm a big plant lover.
Thank you so much for answering all of those questions. Yeah, I do like I am also a bit interesting about biotechnology in these days because um, as I said, I'm in the animal like health and disease like major. So I'm more like I was more towards to more having more knowledge and more interested about like how the biotechnology can be helpful in certain like disease in animals or in something like any research related to like animal field. But like today with your webinar, I was able to learn a bit more about how that biotechnology can be applied in plant, and I'm really glad that I get to listen all of that. Thank you so much for everything.
I'm happy to hear that. And you know, like one of the thing, like I I don't know if you guys are going to high school or CJEP or you're already in university, but I would say for me, going to university was really an eye opener because I've learned about animal science. I've learned about so many different fields of studies, and I think it's important. If you still don't know what you want to do later in life, it's okay. I was there. Now I'm teaching young students about plants and different things. And don't be shy about being questioning what you want to do, being uncertain about what you want to do, because this is normal. Like choosing a field that you don't like just for the sake of choosing quickly and getting in the like work sphere, I don't think it's worth it because you're going to be doing this all of your life and you're going to be evolving. And if you already don't like your field, you're probably not going to like this in 20 years. So try to stick to something you like. Me, I didn't like plant biotech at the beginning, but I liked the idea of finding new alternative, and now I love my field. So it's just a matter of what you want to accomplish. I think it's really important that you listen to yourself. I know it's kind of cheesy, but hey guys, like you're going to be doing that for 40 years. So you better find something you like. If it's plant, then I'm happy. If it's other thing, I'm happy too, but I like plants still. So do you have other questions, guys?
Anyone else have questions for today?
If you have other questions later on that pop up in your mind, just send me an email. I'll be more than happy to answer your question, but also help you if I can. Like I'm telling my students, I'm trying to like make sure everyone get an answer, and if I can help you, redirect you to something a bit more specific or someone I know that knows the answer, I'll make sure it happens. So don't feel shy about sending me an email.
Thank you so much, Dr. Jerome.
Thank you, Chloe.
Thank you for presenting for today. We learned a lot. I appreciate it.
Thank you, guys.